Welcome, guys, to lecture 2. I've added slides, so I will put uh, the updated PowerPoint unnarrated on there, and then of course I'm gonna have to chunk out the narration here on this. I thought I'd just quickly kind of go back over what we discussed Tuesday. Okay, I started out with this slide. Wanting you to move from merely acquainting yourself with getting deep. I think you're hopefully finding that your inquiry is pushing you towards getting deeper in your knowledge there in cell biology. So that was my way to do that. Cell cultures. We talked about the pros and cons of cell cultures, from primary cultures, which come from living organisms. They don't live as long, not even close. You get a limited number of divisions, and that's it. But the primary cultures come from native environments. All the information, the signals, the nutrition, the cell cytokines, the cell signals are all there. The cons, there's a lot of data to tease out, and animal rights concerns. So most cell culture is not primary, it's using cell lines, and that is in vitro, in vitro from glass in culture. It's simplified model systems from different tissue types or organ types. You will focus on parameters independently, so you can control a lot more of the signals, certainly the environment, and you can observe temporal aspects of cells, what they're doing over time. It is not the natural environment, it's an unnatural environment, and there may be signals that are lacking. Okay, but we wouldn't necessarily know that, but at least it's a start. Primary cultures, again, the problem is they're directly from a living organism. They attempt to reflect function from the living organisms, but they will die off. And some cell types, like nerve cells, are in G0, and we talked about the growth cycle, and G0 cells have left the growth cycle. And so they track in a graph number of cells over time, and you see some division, but not very high population, and then they crash. Cell lines are derived from these primary cells. Okay, so they're not clonal, they're actually derived from it. They've been altered. They have altered genes. They are now immortal. They may be spontaneous mutations that occur. They may be from tumor cells that have been isolated from patients. See HeLa cells, Henrietta Lacks' story, good book. Or they could have been virally infected and carrying oncogenes in to a cell. So the virus actually carries in its payload genes are known as oncogenes, and that causes the cells to become immortal. Okay, and you see the graph, a number of cells over time, and you see there's no limit. As long as you keep feeding them, they will keep going forward there. And that's it. Yeah. Okay. Importance of tissue culture, cell culture. Almost all viruses were originally found in animal cultures, except for some that were discovered by molecular techniques recently. Bacteria associated with cells have been found using cell cultures. Making vaccines against things like viruses have been done using cell cultures, and I give you an example: polio, the measles, rubella, chicken pox, etc. And of course, then understanding cancer, the immune system, neuroscience, embryology, development, and fertilization, and the properties of cells after fertilization, the growth. We call that developmental biology. So clearly, that's what cell biologists have, are cell lines, and those are some of the things that have been accomplished using cell lines. Cell cultures, you have to maintain the cells in culture. Again, we're mimicking human conditions, so if they're human cell lines, then they'll be at 37 degrees. The CO2 is 5%. Hey, shh, shh, shh. CO2 is 5%, so we have a CO2 incubator. Depending on the cell type, depending on the media, the conditions, and we can do variation of our growth conditions. So we can manipulate temperature and gas mix, that kind of thing. So that's called empirical, where you come in and tweak the variables to find what works in your hands under your system. You just got, when you're doing a paper, you've got to report any kind of variation from normal procedures. Recipes for growth media can vary in the amount of sugar and growth factors and other nutrients. Okay, right here, growth factors. Sometimes we'll use supplemental media, such as fetal calf serum, and so we don't quite know what's all in the supplemental media. And so we may have to supplement the supplements sometimes. Again, the whole concept is you're trying to grow cells for further study. Okay. Note there is a potential for contamination. You could carry along viruses or prions. So using defined media, defined means that you know exactly what materials are in the media. So there's me spelling defined versus undefined, which would be fetal calf serum. So fetal calf serum, you always see it as FCAS, is undefined. So it's not defined at all. Okay, so you're not quite, I mean, you know enough that it grows, but you don't know necessarily everything in it. So for cell culture, we actually develop and test, study cell lines. It's a permanently established cell culture that will divide indefinitely, proliferate, given media, space, CO2, temperature. Cell lines are different than cell strains. They have escaped the Hayflick limit. That means the limit that primary cells have. Cell lines have gone past that, and they're immortal. Some species, such as rats, mice, give rise to cell lines easy. Some do not. They've never been able to do bird cultures. And cell lines from humans is very difficult, but obviously it's been done. And so a cell line is defined as abnormal, and that is already becoming much like a neoplastic cell, kind of a precancerous cell. So in some ways, even though we call it wild type, what we're studying is not wild type. Primary cells would be wild type. Okay, so these are something different than wild type, but everybody can use them. They're usually a stockpile somewhere in the science authority in the country or the world. Like I said, we have the American Tissue Culture Collection, so it's AT, and you can look up their website, CC. And so you can actually get cell lines from ATCC. So everybody, you know, it's a strain number, and if you do that experiment, you report that in your research, and people should theoretically be able to order the same cells from ATCC and do the same experiments. Some cells need to attach, some do not. So here's without attaching. Okay, these are cells like white blood cells, float in the bloodstream. Others have a surface, cells from tissues, epithelial cells. So if you're growing without attachment, it's suspension. Okay, those that need attachment, the dishes and materials may have to be covered with matrix such as collagen or fibronectin proteins to mimic the organelle uh, structure, and that increases adhesion. And by just connecting and cells layering up to each other actually causes signals for behavior changes. So epithelial cells behave as epithelial cells when they're in a layer stretched across. Okay, they'll behave in that manner. So cell culture is growing cells outside the organism. There's Mr. Mice. And that's a cell dish there that is sterilized. You buy that from the Walmarts. We talked a little bit about pH indicators. You can see the red fluid there. And there's 6 chambers. So you could do 6 different cell lines or 6 different experiments, right? You could add different drugs or materials into each one and then test, does it kill cells or not? So we could take cancer cells and do different levels of a drug and test for destruction. So that means in these little environments, each one's an environment, okay, and that can be observed. So it's hard to tell, but this is plastic, and you can put this on an inverted microscope, and the lens is below and shoots light through. And so you can observe the cells sitting in the bottom there and watch their activity at this point. Okay, watch them grow. Are they alive? Are they dead? And so it's called an inverted scope. Okay, these cells are homogeneous, so there's no other cells to give different signals. You can grow them to large quantities, do many experiments. Lots of different tests and functionality can be done. Non-invasive means we don't have to cut you open and grab cells and study them. Does require specialized conditions, money, tech. You have to hire techs to grow them. Again, there's always this, is this really real? You've taken cells that are immortal, put them into abnormal environments. So it's the best we can do now until we have synthetic biology coming on. And of course, anytime you lift that lid, there goes fungus and bacteria, and you sneeze in it, and there it goes. It's contaminated. Not good. People get cranky. So again, just going back, primary cultures, cells collected directly from tissue, first done in 1907. Cells are minimally modified, but somebody has to die or something's going to be missing. You have to redo it for each experiment because you have a limited lifespan of these, the Hayflick limit, and you're probably grabbing multiple cell types. Okay, multiple cell types. You're not getting one type of cell. When you're, even if a liver, there's going to be blood cells in there and other kinds of material and cells. So primary versus secondary. Okay, secondary is another kind of term for cell culture. Okay, and it's just showing, here's a CO2 incubator. Here it is. You've opened it up. And these dishes here are stackable. And notice the slants here. Okay, so you're allowed a membrane. It's kind of hard to see. There's some holes here. There's a membrane for gas exchange, right? It's oxygen, CO2. Okay, so it could be completely locked in, but it's in a CO2 environment at 37 degrees. So it's heated. Again, the media has the phenol red, pH about 7.4. Okay. Could be undefined media from fetal or horse serum. You add antibiotics to kill any bacteria that you may bring in. Okay. You add glucose, fetal. You add amino acids so they can make new proteins. You add vitamins and other supplements here. Okay. So that's kind of a recipe there. So there is serum-free defined media where we know exactly what's there. Okay, so you start with the minimum and then you add material. It's also known as basal media. Okay, and so you can add material back to the basal media, but you know exactly what you're adding. Okay, so it's kind of like adding a stew. You can add a few things to it, see how it tastes. Okay, you will have to add supplements. These provide certain signals for cells. Mature cells require growth factors, growth hormones, insulin, prolactin, glucagon, other kinds of signaling molecules. Okay, could be other. Sometimes there are special cells that have to grow in with feeder cells. Feeder cells are there in the mix as well. So when we talk about homogeneous cells, that's true unless you need feeder cells. Then you'll have to add a second culture, and those cells release the materials required for the first type of cells to grow. Okay, and this just shows some of the different supplements that have to be added. So this is in some ways very specific for the cells to grow. So it can be very, very variable. Okay. So again, cell lines are not primary, they're secondary. They've been made immortal, they have immortality. Some can be from transformed cells, just a random mutation. Some from biopsy tumors, right? Tumor cells, there it is. There may be specific genes that we add using molecular biology approaches, and we'll talk about that in my molecular in the spring. Henrietta Lacks was the first cells, HeLa cells, from a cervical cancer, which she passed away in 1951. Here's some African green monkey kidney cells, BSC1 cells, 1961. Okay, it's hard to tell, but these cells do look a little different, and if you get further, you would see they have some different structures. This machine actually breaks an organ into cells. Okay, looks kind of sophisticated here. So there are cell harvesters, especially in industry where you're doing a lot of work at one time. This is a machine that disrupts cells by sound. If you've seen a liver, right? I mean, some people eat liver. It's very thick, concentrated bunch of cells. This allows you to take chunks of an organ and break it up by sound waves to release the single cells here for further study in a primary. We have to determine are cells alive or dead, so there are ways to do cell viability. One is called trypan blue, okay, where you can take a sample of the cells out of the dish, put them onto a microscope and add this trypan blue, and you can count the number of cells that have taken the dye up. That means they're dead, versus the cells that do not. It's kind of hard to tell, but they have not taken the cells up. Okay, I'm circling the dead cells here. Looks like this little cell is dead as well. But anyway, you can count the number of dead cells, and so you can change the equations, the supplements, if there's too many of these too quick. Okay. I think I showed this. This is a pathology section, and they have things like lasers that actually come in to a section and flip it out. And actually then you could disrupt this little chunk of tissue, and that could end up being a new cell line. Okay. So here's just different cells. This is under special microscopic lenses and conditions. You can see some cells have a certain structure, others have different structure, and you can tell what's going on based on the structure. This is a laminar flow hood. I believe I talked about a HEPA filter blowing air through these, and this air goes across and then goes below. Here's hard to tell, but this wire, so you're having a constant flow of clean air across this. There's UV light to kill bacteria, viruses. Okay. Those are plasticware pipettes, tips that are already sterile, but you're keeping them sterile by the UV light. Okay, there's other kinds of gas, electricity, vacuum chairs to sit on. Now, this is where you would do your work in a cell culture environment. This is assuming there's nothing toxic, right? If this is just BC level 1, these are just should be very safe. Here's somebody, looks like they're going into some cells. Hard to tell if you see this haze right here. Those are actually stuck cells. These cells are attached, so sometimes they'll take the media and wash the sides, try to wash the cells. There are cell scrapers that scrape the cells like you're shaving your legs or face. Okay, this is some more plasticware. Here's 96 well plate. So 96 different conditions, right? Each well holds about a mil. And depending on the amount of cells and the experiment, depending on the layers and the amount of fluid to be tested, all these are been radiated and sterile here. This is a roller bottle. The whole thing will end up being coated because they turn it like a hot dog on a rotisserie. So the cells stick all the way around. So you increase the surface. With something like this, the cells are only growing on this surface at the bottom. They're not growing on the sides or on the top. With the roller bottle, they grow everywhere. So you can increase the amount of cells if needed. Okay, of course, it costs more money. You have to have a machine to turn these like a hot dog. This just sits flat in a box with some CO2 and temperature. Much cheaper. So less money. Roller bottles, more money, right? More money. Okay, more money. No, you cannot grill your hot dog in the roller bottle. Okay. These are sterile pipettes. They draw up media, fluid, cells. Okay, some have electrical, cause a vacuum to be pumped up. Okay, and then here are some filters that filter media. You add some media here, add a vacuum, and there is a filter that filters out bacteria and viruses, and the suction brings it through. And then you're doing that in a cell culture hood, and then there's a sterile lid you put on top, and now you've got whatever liquid you wanted that's sterile. Okay, so again, cultureware is little plates and dishes where you're growing materials in. Some of these can be coated with different compounds to allow cells to attach. It's just showing some attachment proteins: collagen, fibronectin, lysine, and they don't have any DNA or RNA, so they don't chew up DNA and RNA. They're sterile and non-pyrogenic. That means that they will not initiate any kind of immune reaction. The plastic should be inert to any kind of immune cell. Here are these cell culture hoods called a flow bench, flow hood. Here's some more. Safety cabinet. Okay, it's a little bit more sophisticated than this, but that's the point. Here are the culture dishes. Here's the little scrapers. You reach in. These are sterile, so they come in sterile, like a banana peel, and you peel them back, and in a cell culture hood, and you can then scrape the cells. Okay, here's 96 well plates. They've got 384 well plates. So I could sample 384 patients and test for, say, a virus. I could do one cell line with 384 different types of drugs or conditions and study the effect on the cells. Okay, here's the hot dog roller here. That's what you put the roller bottles on. Okay, he's showing the different roller bottles here. Here's the old world famous scraper. Here's the CO2 incubator. Okay, it has alarms when the temperature, it's kind of hard to see, or the CO2 drop. You know, you don't leave the door open and march around the lab. You close the door. There's two doors here. Okay, and there's incubators. It's hard to tell, but this is actually could shake media. So it's kind of a rotating platform, and it will actually shake. Those are little cell media lids, so you can grow a bunch of cells in a high amount of fluid, and then you can put them into culture or other kinds of 96 wells from the main feeder culture there. These are cryogenic vials, so you can have some cells. If you make your own cell line, mutant cells, you could freeze them, add some glycerol or other kinds of material that makes them survive at minus 80 degrees, minus 80 C, and they can last for years and years, okay, as long as they're kept at minus 80. And here's a box where you drop them in, and organized kind of like a bingo card, so you can actually label on the box top what A1 is, what A2 is, and then you can take a picture, or you can take Excel, and you know exactly box number, whatever, okay, and you keep it organized. You just don't go through every box, right? So you don't want every box out. If it's minus 80, you want to take it out too long, okay? And so that's the first part we kind of covered from Tuesdays. I thought I would stop it here, and then we'll get ready to record the next block of material. Thanks for listening. Let me see if I can stop this.